Illuminating the Druggable Genome is an NIH program which represents an international consortium that has a number of components. These components include, and they're color-coded and you can see the sites, the CEITs, the Cutting Edge Information Technologies, KMC, the Knowledge Management Center. There are 3 DRGCs, that's Data and Resource Generating Centers in kinases, GPCRs, and ion channels, and RDOC, the Resource Dissemination and Outreach Center, which is led by myself, Tudor Oprea, and Stefan Schur, and we do the organizational aspects for the consortium. What is the druggable genome? Well, the druggable genome has to do with a subset of the 20,000 genes in the Human Genome Project, Human Genome, that express proteins potentially able to bind drug-like compounds. And the goal of the program is to improve our understanding of the properties and functions of proteins that are currently not well studied within commonly drug-targeted protein families. So only a small fraction of the genome represents targets, and a lot of the genome still remains currently unilluminated without a lot of detailed information. And so this program intends to illuminate that part of the genome which is not well illuminated. The long-term goals of the program are to advance research, the development, broad dissemination, and use of community scientific resources. And so we're improving availability of information and researching this part of the genome. And then to demonstrate the feasibility and benefits of illuminating the roles of understudied proteins. Eventually, these targets will expand beyond the 3 current targets. IDG is an NIH Common Fund program, which means that it has characteristics of being transformative, exceptionally high and broadly applicable impact. It's intended to be catalytic, short-term and goal-driven, which means that there are data sets, tools and technologies, and new approaches that can be achieved over 5 to 10 years. It's synergistic and enabling. So it's it is intended to be value added to the NIH, with the output enabling the mission of the NIH. It requires a high level of trans-NIH coordination to address complex issues requiring trans-NIH teams, insights, and perspective. And it should be novel, which provides new solutions to specific challenges. The druggable genome consists of these data and resource generating systems, the Resource Dissemination and Outreach Center, and Knowledge Management and CEITs, and also integrates data from external data sources. Our goal is to form partnerships with academic investigators, biopharma partners, foundations, and international groups such as Open Targets. And this meeting is really part of the mandate to build partnerships among academic and biopharma partners. With respect to the components, the DRGCs, the GPCR DRGCs is led by Brian Roth and Brian Choquette at UNC and UCSF. Ion channels is led by Lily Jan and Michael McManus at UCSF. And the kinase DRGC is led by Gary Johnson at UNC. RDOC is, as you see, Stefan Schur, Dushka Vidic, Tudor Oprea, and myself, and Anna Waller, who's the project manager at UNM. And the KMC is there are two components to the KMC, Avi Mayan at yes, okay, and Tudor Prey at UNM. And then there are the cutting edge informatics centers at Georgia and Jackson Labs. And this other one is hiding behind my screen. Okay, so the major sites for contact, we have email contact, which will get you to Anna, important websites, druggablegenome.net and commonfund.nih.gov.idg, and the user interface, Pharos at nih.gov. Okay, so let's talk a little bit about the DRGCs. The GPCR group is led by Brian Roth and Brian Choquette. Their workflow involves in silico docking with zinc compounds for finding probes and genetic approaches with CRISPR and DREAD. These illuminate expression and functionality of understudied GPCRs. And the types of resources that they develop include compound probes, cDNA vectors, and mice. If you go to these websites, you can find the status of illumination of the targets in the protein timeline, and then you can find the resources that have been developed for each of those targets. The ion channel group is led at UCSF. Has a two-pronged approach, tagged ion channels and ES cells for molecular and cellular screening. And they develop antibodies, nanobodies, cDNA vector cells, and mice. You can see at their websites, at Druggable Genome Net websites, the status of their resources and protein timelines. The kinase group uses PRM for profiling phosphoactivity and expression of understudied kinases. They combine these data with systemic and network analysis of the dark kinome. The types of tools that they develop are compound probes, proteomic tools, PRM, peptides, nanobret, and the dark kinase knowledge base. And they also have listings for the progress on their proteins and the resources that have been developed. Knowledge Management Center developed Pharos as the user interface. This site displays information from the Target Central database, and this integrates data sources covering small molecules, mechanisms of action, protein-protein interactions, disease data, expression data, GWAS, phenotypic data of mouse and mouse-human orthologs, data from the literature, harmonisome and ontologies, and all of these sites are of data resources are listed. The cutting edge information technologies, there are three of them. One leverages Reactome knowledge base, a biological pathway database. And there is a IDG-centric portal for this site. The second one develops a comparative genomics framework for visualizing the understudied kinases, and there's an interface for queries for hypothesis generation and knowledge discovery. And you can find out what is available at their site. And then the last one integrates the Monarch Initiative phenotypic data with the TCRD and Drug Central data. And they also expand the NOD2 machine learning tools for elucidating functions of understudied kinases. RDOC provides the administrative support for the program, and we work with all consortium members to collect, curate, and disseminate information regarding critical tools and reagents. And we are involved in outreach to build collaborations internationally. There are a number of tables of IDG resources, which are described: small molecules, nanobret, kits available, genetic constructs, mouse strains, and cells. So one of the outreach activities at NIH is to expand illuminating the druggable genome by R03s. These are small one-year awards that are offered to people who have ideas about unilluminated targets. These dark targets are updated every year and available, described at the IDG website. So these investigators, and you'll hear from several of them tomorrow, each year explore a series of targets in competitive programs. In the first year, there were 16. In the second year, there were 17. And these targets span the interests of illuminating the druggable genome target families, kinases, GPCRs, and ion channels. And the third round of competition is currently going on. NIH also funds related druggable genome awards. In particular, there have been administrative supplements for identification and validation of new pain and opioid use disorder targets within the understudied genome. And there are about 8 of these, 9 of these projects for novel unilluminated targets that are relevant to the HEAL initiative. And IDG is also associated with SBIRs and STTRs, which have to do with commercialization of results related to understudied proteins. And then there are also resources for utilization of IDG, including early stage preclinical validation of therapeutic leads of interest to NIDDK. Finally, Tudor Oprea has organized a series in Nature Reviews Drug Discovery called IDG Target Watch. And this series, which has the potential of being monthly, now represents updates that are coming out of the IDG program across the protein families, as well as general perspectives on novel drug targets each year.